These two chromatids are originally two linear molecules but packaged together through the use of histones. Let's pull these two strands away from the chromatids. This chromosome now became two linear molecules of DNA. Remember that DNA is double helix, but for convenience sake, we'll make them a line from now on. So here we can clearly see the VDR gene after the chromosome was laid out flat. We'll now look further into what this gene is made out of. First, we have a promoter site, which is where RNA polymerase II binds to and begins transcribing the DNA. Sometimes the promoter needs a DNA sequence, TATA, or they call it TATA box, to recruit an enzyme called TATA binding protein. Once the TATA binding protein binds to the TATA sequence, the entire helix will bend to allow transcription factor and RNA polymerase II to assemble. Once this happens, transcription begins. In the promoter site, there may also be something called an enhancer that acts to regulate transcription, but depending on the progression of the project, we may not need to keep this in mind. The middle is where the genes get transcribed into RNA. Notice that both exon and intron gets translated, but before mRNA leaves the nucleus, RNA processing occurs where the intron gets spliced out, exon knit together, and poly A tail added on with the exon. It then goes out of the cytoplasm towards the ribosome. At the end is the termination sequence, where the RNA polymerase II stops transcribing. If we look at Figure 3 of the review paper given to us, we see that there is a promoter region, and within the genes that will be transcribed, DNA binding domain from exon 2 to exon 4 and ligand binding domain from exon 6 to exon 9 are also involved. So what are they? Ligand binding domain is a part of the protein that can bind with hormone. In this case, part of the VDR gene protein transcribed from exon 6 to exon 9 binds to 125-OH2D or vitamin D. Take note that hormone includes steroids, and steroid looks like vitamin D, so hence ligand binding domain can bind to vitamin D. DNA binding domain is basically a protein domain that have high affinity to bind to specific genetic sequence. What it means in this diagram is that part of the VDR gene transcribed by exon 2 to exon 4 will have a VDR peptide that contains a DNA binding domain. This DNA binding domain have high affinity to bind to VDRE, vitamin D response element, which we'll talk about in a minute. This transcription of VDR gene will eventually transcribe a VDR protein with ligand binding domain and DNA binding domain that binds to vitamin D and vitamin D response element. Let's call this VDR complex. Vitamin D response element is a short sequence of DNA within the promoter of a gene that is able to bind to specific hormone-related complex, or VDR complex. Now here's the biggie. Vitamin D response element is a sequence that exists in many different promoter sites, at least 200 genes, which means that once these vitamin D response element binds to VDR complex will regulate transcription of many genes that contains promoter with vitamin D response element. In other words, the binding of VDR complex to VDRE will either upregulate or downregulate the genetic information of at least 200 genes. These genes are ones that will be regulated and thus induce cancer. Now let's take a look a bit deeper into some of the concepts introduced in this report. Remember that if you inherit a specific allele of FOK1 and BSM1, the chances of having breast cancer increase. This is the main idea of the article. We'll look at what allele means in a second. If we look at figure 3, we see that FOK1 is in exon 2 and BSM1 is in the intron between exon 8 and 9. FOK1 allele is basically a sequence within the VDR gene that can vary by one base pair due to polymorphism. Remember that single nucleotide polymorphism is when one base pair is either substituted, deleted or added due to mutation. In the case of FOK1, it is substituted at exactly position 30875 from a cysteine to a thymine. Let me explain this position. Imagine a VDR gene transcribed into an mRNA that will subsequently be translated into VDR protein. In the process of mRNA translation, we will hit a code called an initiator ATG sequence, which translates into MET protein. The beginning of the protein translation, if you remembered. A in the ATG represents 1, T is 2, G is 3. From that place onward we count, and we find position 30875. That is the position with the substitution. In fact, the small f allele means the sequence has been mutated from the original cysteine to a thymine, somehow causing 3 extra amino acids inside the VDR gene. This additional amino acids in the VDR protein explains the different risk factor depending on which 2 F alleles we have. Big F allele means this position has not had the substitution and remains the same. Since the 2 F alleles work in a codominant system, it works together to produce the same protein. If both Fs are non-risk factor allele, then we have 2 copies of the gene working in your favor. If both Fs are risk factor allele, as with the case of breast cancer risk, and the 2 small f alleles, then both alleles are not in your favor. If we have a big F and a small f, then one doesn't work in your favor while the other tries to compensate for your risk-prone gene. This is the dominant system. This is the codominant system, where alleles work together to handle a specific function. When one team member is defective, the other takes over and tries to do the work, but may have compromise in performance. In the case of BSM1 allele, it is also working in a codominant system. A small b causes risk for breast cancer due to mutation in position 64922 of VDR gene that substitutes a guanine to an alanine. BSM1 is in an intron close to the terminator sequence. In other words, it is close to where poly A tail is processed. In fact, 2 small b alleles for BSM1 is linked to longer poly A tail, disrupting the stability of the VDR gene from transferring to the ribosome for translation. Allele. Remember we have 2 chromatids 12, or 1 single chromosome 12, and that one is inherited from mother's egg and one from father's sperm. We entangled the DNA into 2 molecular lines and found 2 transcribable template strands. To tie things up a bit, each copy of the big F or small f allele is in each one of the chromatid, and the type of allele we have depends on what our mother and father initially possess. This single nucleotide polymorphism is not a mutation that occurs in our lifetime. It is likely the mutation of our long history of ancestors. We may have different chromosomes from each of our parents, which translates into 2 different copies of, say, small or big F allele. These alleles variations are a result of single base substitution that deviates from wild-type alleles. This is the gist of pretty much what the article encompassed.